What's up, guys? It's your non-gendered cell, Kai, coming at you with this explanation of me and my cell cycle. So, to summarize my existence, there are 2 phases and 5 steps to creating a bunch of inferior versions of myself. So, we're going to start off at G1, where I begin to develop, differentiate, and grow. And I realized, dang, my surface area to volume ratio is out of whack, and I do be looking thick. Guess I gotta either commit apoptosis, split in 2, or go to G0, and literally do nothing with my life. I've decided to continue, and now I'm committed to completing the cell cycle. So I go into S phase. In S phase, I copy my DNA, and for each homolog, it attaches to the sister chromatid at the centromere. So now I have 2 chromosomes with 2 chromatids on each. A good way to remember how many chromosomes I have, in case you can't count, is by counting the number of centromeres. After S phase is G2. In G2, we prepare to divide and we duplicate all of our organelles, make proteins, and store food. So now I have to stop at this G2 checkpoint gate, and here I have to check for DNA damage using proteins like P53. And if we somehow mess up our literal only job, we try to fix the damage, or we just go commit apoptosis and bleb. Whoopee! Now go into prophase 1. Here my centrosomes, which are made out of centrioles, will duplicate and start to move to the opposite poles of the thick cell, and my nuclear membrane will also break down. My chromosomes will condense and become visible. Homologous chromosomes will stick together in pairs in what is called synapses to form tetrads. The chiasma is where our non-sister chromatids will meet and where crossing over will occur. Have you already forgotten what crossing over was because you procrastinated at 3 a.m.? Well, crossing over is when our non-sister chromatids exchange parts of their DNA with one another at the chiasma point to help create more genetic variation. I now reach metaphase 1. In metaphase, my centrosomes migrate to opposite poles because they hate each other, and my spindle apparatus forms and attaches to the chromosomes at the centromere. The tetrads begin moving towards the metaphase plate and eventually line up along it, while each of our duplicated pairs of homologous chromosomes attaches to a microtubule. Our spindle apparatus is formed from spindle fibers called non-kinetochore microtubules and kinetochore microtubules. Now I've reached anaphase 1. In anaphase 1, my pairs of duplicated chromosomes separate and start to move towards opposite sides of the cell. I begin to elongate as microtubules push the poles of my cells further apart, and my duplicated homologous chromosomes are now at separate ends of the cell. My non-kinetochore microtubules will lengthen to elongate the cell, and my kinetochore microtubules will shorten to pull the chromosomes. At telophase 1 in cytokinesis, an actin filament will split me in half by the middle and form a cleavage furrow. Cleavage. And the chromosomes will uncoil. Now, single chromosome sets are at the very far opposite sides of my cell. Nuclear envelopes will reform and cytokinesis occurs along the midline of the cell, separating it into 2 cells. Our cell membrane diploid cells with 1 egg, 2 eggs.
Now we repeat the process, but a little bit differently. Centrosomes will start to move to the poles of the cell, and the nuclear envelope breaks down again. The chromosomes are still in 1N, 2X form, shaped like an X, but they won't stay like that for long. Now I head to metaphase 2, where the meiotic spindle starts to form and attach to the chromosomes at the centromere again. The chromosomes will begin moving towards the midline, or equator, of the cell, called the metaphase plate. Chromosomes will be lined up individually along the metaphase plate, where each sister chromatid will be attached to a microtubule or the kinetochore of a microtubule. Next step is anaphase 2. Sister chromatids of each chromosome separate and move, and each sister chromatid, now separate chromosomes, is at the separate end of the cell poles. Last off, telophase 2. My nuclear envelope reforms and the spindle disappears away. The cleavage furrow will form again and the chromosomes will uncoil. A division occurs along the midline of the cell, separating each cell into two cells. The nucleus will reform after cytokinesis, and we will get 4 haploid cells with 1 N, 1 X. In females, meiosis, which is called oogenesis, will produce 4 haploid cells, but 3 will become dysfunctional polar bodies, and the other will be an egg or an ovum. In spermatogenesis, the body will create 4 functional sperm cells with DNA sets each.